a deepfake-based approach to generate radiogenomic data in cancer. Radiogenomics. You might be wondering about the meaning of this term if you haven't heard of it before. Radiogenomics is the combination of two terms, radiomics and genomics. You should have heard of radiomics, which refers to medical imaging, and genomics, which focuses on the structure and the function of the genome. From this background, radiogenomics has arisen, as it's a newly emerging field that integrates radiomic and genomic data to elucidate the association between gene expression data and imaging phenotypes to serve the medical studies, especially in cancer studies. Radiogenomics is a promising field, but it's hindered by the unavailability of numerous big data sets of paired imaging and genetic data for the same subjects that are crucial for training machine learning-based techniques utilized in the analysis pipelines of radiogenomics studies. This is a problem. How can it be solved? After searching for a solution for this problem, we propose a deep learning-based approach to generate data that are hardly differentiable from realistic data to solve the data unavailability problem. This approach serves the healthcare facilities and the researcher community by providing very large data sets to suit the training data requirements. The expected output is a deep learning-based model that generates either or both of the two types of the radiogenomic data, aiming to build a large dataset. To apply this deep learning approach, we plan to use the technology used in deepfake applications. After a lot of search on deepfake tools, we have come up with GAN. But what is GAN? GAN, which is a generative adversarial network, is a class of machine learning frameworks that has two neural networks contesting with each other in a zero-sum game.
Now we are going to talk about our project progress. We started our journey by trying to understand the core of the radiogenomics as it focuses on the relation between the imaging characteristics of a disease imaging phenotype and its gene expression patterns genotype. And we searched for techniques used in data augmentation and we come up with generative adversarial networks. We moved to understand the basics of GAN, its history, components, different types, and how it works. As we said that it consists of two neural networks, the generator, which has two inputs, the latent space, which contains features related to real data, and an n-dimensional noise vector. Then the generator generates fake data, and the discriminator, which has also two inputs, the real data and the generated data, and it tries to differentiate between them. Then it sends its feedback to the generator to tune its parameters to generate new fake data that is closer to the real data. Then we plan to start our journey with understanding the basics of deep learning by studying the deep learning specialization courses offered by deeplearning.ai on Coursera. This specialization was a great beginning for our journey as we learned from it how to build, train, and apply fully connected deep neural networks for classification problem using vectorization technique. How to optimize our model using algorithms such as mini-patch gradient descent, RMS probe, ADAM, dropout, rather than using patch gradient descent. We also learned how to build a successful machine learning project and practice decision making as a machine learning project leader, how to build convolutional neural network and apply it to visual detection and recognition tasks. After that, we reached an important station in our journey, which opened the door for us to dive deeper into a real implementations for the GAN model. We reviewed published papers deploying GANs for applications in medical imaging and genomics studies. In our literature review, we worked on understanding different deepfake techniques for generating realistic gene expression profiles and medical images. The first paper, Generative Adversarial Network for Medical Images (MIGAN). In this paper, the authors introduced a new Generative Adversarial Network for Medical Imaging (MIGAN) framework for generating synthetic retinal vessels images and their segmented masks from only tens of examples. In their proposed technique, they update the generator twice then the discriminator to get quicker convergence to reduce the overall training time. The second paper, Adversarial Generation of Gene Expression Data. In this study, the authors addressed two problems. First, they proposed a mechanism to assess the realism of a synthetic gene expression dataset. Second, they managed to design an adversarial simulator of expression data using WGAN-GP architecture. We managed to reach the same results in both of the papers.
Now, we have reached the most interesting phase in our project. We started to download our project data using TCGA for gene expression data and TCIA portal for imaging data, and we collected our data for glioblastoma multiforme disease. These two portals provide paired genomic and imaging data for the same subjects. Let's start to talk about the technology used in our project. After a lot of search to understand the GAN architecture, we faced a very big problem. GAN often suffers from training instability. Based on that, we searched for an improved version of GAN, and we found WGAN. WGAN makes progress towards stable training of GAN, but sometimes can still generate only low-quality samples or fail to converge. These problems are often due to the use of wet clipping in WGAN. After a lot of search, we found an improved version of WGAN. This version is WGAN-GB, and it performs better than the standard WGAN by introducing gradient penalty and enables stable training of a wide variety of GAN architectures with almost no hyperparameter tuning. And we were able to build this architecture using Python programming language with TensorFlow 2 on PyCharm IDE. TensorFlow is a free and open source software library for machine learning developed by the Google Brain team.
Now we are going to talk about the innovative aspects of the project. We will incorporate correlated features from the gene expression data and the imaging data to train the GAN model, not only features taken independently from either of them. These features link a disease phenotype with its genotype. Applicability. This project could be the foundation for a software product that can accept one type of genetic data or one type of imaging data and generate many other genetic and imaging data types. Social and industrial impacts. This project can lead to reduce the cost for a medical research by cutting the budget needed to undergo genetic and imaging tests for a large number of subjects, which can help in understanding the underlying mechanisms of how diseases such as cancer develop, and improving diagnosis precision, prognosis assessment, survival estimation, and treatment response prediction.
Deepfake technology is a double-edged sword that can be used in either a constructive way or a destructive way. And when it is used in a constructive way, it provides very useful applications in the scientific research era. Thanks to this technology, data augmentation became a real process as a solution for the data unavailability problem that hinders the scientific research community. Thank you for watching.